Here we go with part 2 of our protein synthesis lecture. Remember last time we talked about transcription, which is taking a piece of DNA and making a complementary mRNA from it, okay? And here we've got a picture of a eukaryotic cell here. So ours our DNA, make an mRNA, then it gets all processed, it's mature, then it's going to exit our nucleus, go to the cytoplasm, and now it's going to get translated into a protein with the help of ribosomes and tRNA. Okay, so again, last time was transcription, turning DNA into mRNA, and this lecture is going to be now talking about how we turn mRNA and we translate it into a protein. How does the protein actually get made? Okay, so let's get into it. Again, chapter 14.4 in your book if you're wanting to know where you can find this or follow along. Translation has 3 parts: initiation, elongation, and termination, which are the exact same steps as transcription, right? Same names at least, right? We're going to find the process is different, okay? But as you can imagine, initiation is the start, elongation is we're building it, and termination is the end somehow, okay? So with initiation, the 5' end of the mRNA that has that little cap on it, it is going to interact with rRNA, that's ribosomal rRNA, okay? And basically the ribosome is first in a couple different pieces, and when like the first piece kind of bumps into that 5' end, that's going to attract the rest of the ribosome to it, and so then the ribosome is going to kind of assemble around the piece of mRNA here. And so this is going to happen right at the kind of beginning of the gene that's going to start coding for an actual protein. And this beginning area is called the start codon. Okay, so initiation is the ribosome assembling around the mRNA near the 5' end. And then with elongation, this is where we are actually building our protein, amino acid by amino acid. So we're taking a monomer and we're linking it together to form a polymer, okay? And so basically what happens here is this like GCA, right? This might be 3 RNA nucleotides in a row, and that's called a codon. And this codon is going to code for one specific amino acid. Okay, so if I look over here, GCA, first letter G, second letter C, GCA, that codes for the amino acid ALA, which I think is alanine. Okay, so this would code for alanine. Notice that all of these codons code for alanine. So there are many different letter combinations that code for amino acids. This chart here shows all the different amino acids. There aren't that many, right? And so you would never have to memorize this, okay? But you'd have to know how to use it. That if there's a GCA on the RNA, this would lead to an alanine amino acid, okay? So what happens now is our ribosome goes along and it reads this triplet, okay? And so what's going to happen is this triplet is, again, this codon is going to code for a very specific amino acid found on here, okay? A tRNA, a transfer RNA, is going to bring that specific amino acid. So the first codon is always AUG. This is the start codon, okay? But AUG, if we look it up on here, AUG starts for met, which is like methionine. So all proteins start with methionine, okay? So a tRNA is going to come into the ribosome, and up here is the amino acid. Here's the methionine. So this tRNA brings this, and it has a complement that matches up, and so it says, oh, yep, I'm the met tRNA. Yep, this matches up perfectly. So here's the methionine you wanted. Okay, and so it's first going to come in through this A zone, which is the approach, okay, and then the amino acid is going to move a little bit, and then it's going to move to the P zone, and then finally it's going to leave through the E zone, which will make more sense in just a second, okay? Okay, so the amino acid comes in through the A, so comes in, okay, and then here's our next codon, okay, so here's our next tRNA coming in, bringing the next amino acid, okay, and they are going to, these two, the MET and the PHE, are going to get attached together in the P site of the ribosome, Okay, and now this guy, it's met as attached to the PHE, so this guy now leaves E through the exit site, and now our next one's going to come in, and then this arginine would be attached right here, and then this guy would move on and so forth. Okay, so we're going to show a bunch of different ones here. So here we go. This one's coming in. This one just left. Okay, these two are now getting bound together, and next this one's going to go. Then this one's going to come in, and so on and so on. So we keep just bringing in a new tRNA that has a codon that is complementary to this codon, and each tRNA has one specific amino acid that it carries. Okay, tRNA are in this kind of funky shape, okay, where the amino acid that they carry goes up here, and then their anticodon that matches up with the RNA. So like here, this AAA would be the codon, and the UUU is the anticodon. Okay, that's the codon on the tRNA. So here is the anticodon down here. So that's the part that matches up with the mRNA, and then up here is where our amino acid is. Okay, and it's in this crazy loop little structure here, kind of. Okay, it's really kind of like this 3D, but it's in this crazy structure. But the idea is the anticodon is down here, and the amino acid is way up here on the other end. Again, here's just one more picture. Okay, this guy just came in the A zone, and now these two are going to get attached together. So now notice. Now, boop, we move down. This guy's now in the E zone, exit, it's done. Now this one's here, the next one's going to come in, and the ribosome is going to kind of march down the mRNA, and so on and so on, and we'll get another tRNA coming in and another one leaving once its attachment is done. So that's elongation, and now the final step is termination. And how do we know? There is a specific stop codon, actually, okay? And so this stop codon is UGA, codes for stop, okay? You see right there, that's what that stop is, okay? So when the ribosome gets to that UGA codon, and actually there could be a couple, when it gets to one of those stop codons, then what happens is instead of tRNA bringing an amino acid, a release factor comes in, and this release factor is going to pop open our ribosome, okay, and it's going to release our polymer right here, okay. And so here it is all together. Initiation is where one little piece of the ribosome meets up with the mRNA near the 5' end that attracts the big piece. Then our tRNA are going to start bringing the appropriate amino acids based off of the codons and anticodons. And then when it hits that stop codon, a release factor is going to pop it off and release our protein. Good news for you, unlike last time where transcription was different in prokaryotes and eukaryotes, it is the same. Translation is the same. Both happening in the cytoplasm, both happening in the ribosomes. There's no difference between prokaryotes and eukaryotes with this process. And so as it turns out, this whole idea of DNA then leading to RNA, and then RNA leads to proteins is like a central tenet of biology. It's like a foundational principle. It's a really big deal. So let's go back to first semester now. If we're making a protein, I want you to think about, is this going to be anabolic or catabolic? Well, we're making a polymer, right? So that's going to be anabolic, right? Anabolic, catabolic. Androgonic or exergonic? Think about it. We're making bonds here, which means we're going to require an input of energy. So it's going to be endergonic, input of energy. And hopefully you can remember that because we're building a polymer, this is dehydration synthesis happening inside the ribosomes. Okay, that's how the amino acids get attached together. So here are the overall steps with both translation and transcription, some of the main ideas of what you should take away from this lecture. We're going to be doing some hands-on with this. We're going to be working with it more in class, but hopefully now you feel pretty well equipped and you've got some of the vocab and some of the visuals that you might need. And again, in the textbook, this is chapter 14. Check there for more explanation, more visuals, etc.